Hello, everyone. My name is Stephanie El-Hayek, and I'm the Assistant Director at the Center for Arab Genomic Studies. And I would first like to welcome everyone to the second webinar, part of the CAGS webinar series. This webinar series is being organized by us at CAGS and our parent organization, the Sheikh Hamdan Award for Medical Sciences, with support from the Dubai Health Authority, the Ministry of Health and Prevention, as well as the Emirates Medical Association. As many of you know, last Wednesday, March 24th, 2021, Sheikh Hamdan bin Rashid Al Maktoum, the patron of our organization, passed away. Sheikh Hamdan was a true visionary who dedicated a lot of his time and effort to improving the healthcare here in the UAE and also around the world. In fact, it was his vision of reducing the burden of genetic disorders in the Arab world that led to the establishment of our center. His passing is not only a loss for us at CAGS and the award, but also for the wider medical and humanitarian community. We truly hope that we will be able to continue working down the path that was guided by Sheikh Hamdan's vision. I would like to now hand over the screen to our moderator for this session, Professor Riyad Bayoumi. And I hope you will find this session and the upcoming webinars in our series insightful and beneficial. Thank you.
Thank you, Stephanie. My name is Riyad Bayoumi. I'm the professor of biochemistry at the Mohammed bin Rashid University in Dubai. And I'm going to sort of look after the webinar on behalf of the Center for the Arab Genomic Studies on population genetics. We have two speakers today, and I will introduce them one by one. But let me first of all introduce some housekeeping sort of notes. First of all questions must be written in the question and answer box, and they will be answered at the end of the session, at the end of the two speakers. Then we will open the questions and answers session. CME certificates will be downloaded as of tomorrow from the link which is on the screen, and you will receive notification of that. And also, we appreciate if all the people who are attending the webinar could please answer the short survey at the end of this webinar. Now, the first speaker is Professor Pierre Zaloa. He is the professor of biology in Khalifa University in Abu Dhabi. He has been in Lebanon previously in American University of Beirut, and he has also been an adjunct scientist at the Chan Harvard School of Public Health in Boston. And he has been involved in population genetics, and in particular, his contribution to the Genographic project, which has been looking at the history of human migrations over the last 100,000 years. The title of the talk of Professor Pierre is Exploration of the Ancestral Genetic Landscape of the Arabian Peninsula and the Levant. So may I request Professor Zaloa to start his presentation.
Greetings, everyone. Thank you for the invitation. I'm very honored that I was able to participate in this, series of webinars. Hopefully I can stick to my time limit and discuss this topic that is very dear to my heart. And without a lot of delays, let's start this journey. So before I talk about the topic, which is how people moved into the Levant and the peninsula, which is a very defined region, let me just, for the first two slides, make some important observations that we've learned over the last 15 years. initially, we never anticipated, as a matter of fact, when I was teaching my student genetics, we always told them that humans, modern humans, never admixed with archaic humans, namely Neanderthals or any other human that left Africa earlier. But again, things have changed with the advent of ancient DNA work. And now we know for a fact that humans or modern humans or Homo sapiens sapiens interacted with the Neanderthals not once, not twice, but at least 4 times so far that we know of. And not only that, but we know also that modern humans admixed with another group, which we call them Denisovan, because of the Denisova cave in Asia that's where we discovered. So basically, the story that we have now about human migrations has been a bit spiced up with the fact that we are, as modern humans now, we have a touch of Denisovan DNA and Neanderthal DNA in various degrees, depending on the locations. So we not only know that we admixed with Neanderthals and Denisovan, but we know roughly the time of admixture, and we also know where these admixture happened. We know that in Africa it didn't happen. So soon after modern humans left Africa, they interacted with the Neanderthals, although there is a very recent study that is debating the date. It's pushing the date a bit backwards. Instead of 25,000, perhaps it's 40,000 years. But in any case, it happened somewhere in the Levant initially, and then later on in Asia, as you could see on the top right of the slide. And also moving to Asia, they also admixed with the Denisovan. So Asians will have Neanderthal DNA admixed as well as Denisovan DNA admixed. And people in Eurasia and the rest of the world, except Africa, will have Neanderthal DNA. What we know also is the fact that this told us a refined story of how humans moved out of Africa and the directions they went to based on these archaic admixture. So as you could see from the part on the right, these are the admixture events and the dating of these admixture events. And we can at least have 5 of these. These slides were taken from Nature publications and Nature reviews. The rest of the talk will be my own and my team's slide. So having said that, now we have to really understand that when we talk about human migrations out of Africa, I'm always referring to the people who actually are admixed with the Neanderthals and how and we follow on that part onwards. So what has been established over the years using DNA evidence is the fact that there are 2 routes of migrations out of Africa. And when we talk about the out of Africa migration, we are referring here to the most recent and most successful out of Africa migration. There has been many out of Africa migrations in the past, but these did not actually lead to a successful establishment of Homo sapiens sapiens outside Africa. So here I'm referring to the most recent and most stable, if I could use the word stable, migration out of Africa, which happened based on DNA evidence anywhere between 60 and 40,000 years, more likely 50,000 years. And this evidence, DNA evidence, is supported by initially mitochondrial DNA, then Y chromosome DNA, and now with ancient DNA and autosomal DNA. So we are almost certain about the dates of the out of Africa migrations. And of course, the stepping stone outside Africa is the region of interest that we are talking about today, which is the Levant. If we talk about the northern route of migration through the Sinai Desert, which was not a desert back then, into the Levant, and then the other route, which is Bab el-Mandeb route, or the Gate of Tears, through Yemen into Arabia, and then further north. So I will dissect those events using modern DNA, some ancient DNA, and supported by archaeological evidence and other evidence that I will share with you. So when people migrated out of Africa, let's say around 50,000 years ago, the climate was favorable. It was not favorable in Africa, and that's why they left Africa. But when they got to this region outside Africa, soon after that, the climate changed. And when the climate changed, sorry, I missed the slide here. So when the climate changed, basically, we know what happened during the last glacial maximum, which lasted, a couple of decades and a bit more. So it left the entire region, Southwest Asia, which is, from parts of the Levant to the peninsula, left it arid, that cannot support human life or animal life or even plant life. And this was supported by archaeological evidence, because we don't see hard evidence of human remains that actually are within that period, less than 40,000 years to about 20,000 years. So it's very scarce, and we think it's because of the climate. It was not supportive of that region. So what may have happened, and this is the hypothesis that I have proposed, is that people left this region and went again searching for refugia where they could find food and animals to hunt, etc. And these pockets or refugia, they were found around the Black Sea region, as we know from maps that actually are extrapolated based on climate maps, as we call them. So you could see on the slide where you have the green spots. These are areas where they could support life, and perhaps in East Africa as well. So what may have happened during this last glacial maximum is that these humans that actually lived there migrated and escaped into these. And they stayed isolated for a very long time until the ice melted. And once the ice melted, then these people or these small populations came back into this region. And that's why I'm proposing a post-LGM migration routes. So post last glacial maximum, and they use the same routes again. One from the north through the Sinai, and one from the south, which is through Yemen or Bab el-Mandeb or the Gate of Tears. So these are the folks that actually went into East Africa after the ice melted, or during the last glaciation, and went back. And of course, the same thing happened to those who actually inhabited these verdant areas or refuge across parts of the Levant and across the Black Sea, and they went back populating the entire region. Now, of course, this was the hypothesis that we have, and we tested it using DNA. And we initially worked on Y chromosome DNA. It was simpler, it was available, and we started working on these. And we basically looked at population expansions using Y-DNA as a model. And indeed, what we discovered is we discovered centers of expansions based on these DNA findings, and they are consistent with the hypothesis where refugia around the Black Sea, parts of the Levant, and also part of Africa, which I don't show here on the slide, but take my words for it, we have these expansion centers. And not only we have these expansion centers, we have dates on when the expansion happened. And these dates, if you look to the left part right here, we're talking post glaciation. So we're talking 13,000 years and then 12 to 13,000 years as well, when these folks started to expand. And it's very consistent with our knowledge of how archaeological evidence that started to expand from these refugia. And of course, to the right, it's not of our interest today, but we also show the expansion from these centers, not only to the area of interest, but also through Europe as well, which happened a bit later. If you could look at these maps, as a matter of fact, we did not draw them just based on where we would expect to find people. This is really where populations were and how expansions happened based on DNA evidence. So it was really shocking to us where we could lay this on a physical map and discover those expansions. So this is I'm just checking the time here. So basically, we expanded this a bit, and it was consistent. We did additional Y chromosome DNA data, and then we were consistent with these expansions and source of expansion. So when I have a dot right here, this means this is a source population. Again, from it radiated smaller populations, expansions, consistent with the previous finding and consistent with our hypothesis. When we plot the cultural timeline and the climatic timeline, we have a sync between DNA findings and cultural adaptation and also archaeological remain. So unfortunately, you don't see the very clearly what is meant by the orange dot. I lost it on the slide. The orange dots, they represent evidence from archaeological evidence of human remains. And of course, the woodland area are represented by green and plant refugium by blue, and they were consistent exactly with our DNA findings and consistent again with archaeological remains. And as the climate got better and better, and as these cultures started to appear, we see more expansion, and we can actually map the entire Fertile Crescent, actually, that being physically starting to appear to take that shape based on these archaeological remain and DNA evidence through these routes of migrations. So that was all good. And then the early cultures that were actually there supported these evidence. But we wanted to probe this a bit more and figure out, was there really a story that we can tell about these cultures and relate them? So these cultures that appeared 15 to 14,000 years ago, can we relate these cultures to the modern inhabitants of this region? And that was the second phase of the project that we started. Of course, just to show you here that indeed we have also shown the expansion to Europe, but it's not for the topic today. So let's delve into the recent study. So we did a whole genome data analysis on these, and we used ancient DNA samples from prior studies published, and we used other populations that we actually collected. And the next speaker will also be talking about some of those, because we work together on these projects. And basically, we have close to 1,000 samples collected from the entire region of interest, and of course, around the region as well, because we wanted to have some, a bird's eye view on what could be happening. So the first thing that you would look at, and don't really try to figure this one out. I will show you a next slide with an easier image that you could tell from. But I wanted to show you that we've done it across many populations. A lot of them you can't even read here, but we've done it for the entire region that you see on that particular slide. So what did we get from this? What we got from it, when we compare Arabian Peninsula, and by Arabian Peninsula, I mean the entire peninsula, including Yemen, we find a difference between Arabian Peninsula and the Levant, but we also find a lot of similarities. And for example, we were able to show that there are at least 6 ancestral components that actually are present to a various degree between the Levant and the Arabian Peninsula. Natufians are present in both in a dominant way. Neolithic Anatolians are differential. In some parts of Arabian Peninsula, you'll find them, but not in other parts. Neolithic Iranians as well. Ethiopians and Somali, these are the back migrations that I've talked about. And of course, we have the Gujaratis in India. There was a lot of interaction. Most recent, when we talk recent, we're talking 6,000 to 5,000 years prior, and some from the Pontic steppes. And I'll go through these in further details. But what was also interesting, if I can move this, maybe, yes. Okay, so what was also interesting is on a PCA, we were able to see some differences or some demarcations between Arabian Peninsula and the Levant, and clearly we could see here if we look at the blue, that's the basic Levant, and the peninsula is spread around closer to Yemen. And these would be the Neolithic Iranians. So the Levant is smack in between, which is it's something that we expected to find. So if you look here more clearly, the Levant to the left and Arabia to the right, so you could clearly see some differences and some affinity. So the Levant shows a tight cluster between, as I said, the Arabian Peninsula and the Persian Gulf. And the Arabian Peninsula shows a cluster, which is the Yemeni cluster, which involves the Persian Gulf region, which got us to think about certain possibilities. So we did other, analysis to identify the route of expansion, some basic F4 stats. And I don't want to go in details through this, but basically you take an outgroup, which in this case is the chimp, and you take an ancient population, and in this case Neolithic Iran, and you want to see throughout which you test. Let's say you are testing a population, let's say Jordan, and you want to see if there is a link throughout this migration pattern between Jordan and Yemen, and if it goes within a positive line, then there is a continuum, and if it goes the opposite, then there is no continuum. I don't want to confuse you, as I said, so let's skip that and get to the results. When we look at the results, so what we have shown is that there is positive sharing with ancient population. So what came through Yemen, for example, through Neolithic Iran, so there is a positive sharing, which means that, yes, so what happened is if you go out of Africa, that route, you go through Yemen and through the Neolithic Iran. So there is a continuum. So there is some sharing of genetic evidence. And again, negative values, meaning that there was no sharing. So modern Arabian populations, they harbor ancestral lineages that are probably of African origin. And I will dissect this in a minute. So what we found out from basically the admixture results, the PCA, the F4 stats, and the graphs that I just shown you is that the lineages from Yemen to the Neolithic Levant and the Natufian do not positively correlate. So basically, people that moved through Yemen did not go to the Levant. Okay, but what we see as well is correlation of Arabian lineages with Iranian genetics. We see that, but not for Neolithic period or earlier Levantine genetics. What does this tell us? Is that expansion into Arabia passed towards Iran and not the Levant. So this is the first basic summary that we can get from this. The second summary is that the split that occurred, if you remember about these routes of migration, it occurred early enough that differentiation must have evolved between the Neolithic Iranian and the Natufians, because modern Arabians do retain some lineages with the Levantine lineages. And these are from the Natufians that we saw earlier. And the Natufians and the Neolithic Iranian lineages, they are differentiated, and then each one went a separate way. One admixed with Iranians and one admixed with the Yemeni, which basically got us to identify expansions and distinct evolutionary path that were considered. Now, if you look at this summary paragraph, you will get scared, but I'll walk you through it in a very simple way. I think I may have a summary of it in the next slide. But just to let you know, so if you look to the left, there is the Levant, and if you look to the right, there's Arabian Peninsula. And all I want you to focus on is 3 arrows right here, same 3 arrows right here, and these are Neolithic Anatolians, Natufians, and Neolithic Iranians. And these are the ones that actually make up the entire Levant. In Arabian Peninsula, while you have some Neolithic Levant in certain parts, the majority come from Natufians and Ethiopians. What I mean by Ethiopians is some East African parts. And in certain parts of Arabia, you will see Neolithic Iranians. So how do I summarize all of this? In the Levant, and this is post-last glacial maximum, okay? We're talking here, you know, I mean, these dates now vary a little bit, but they are less between 12 and 6K, the majority of those. So we believe that the Neolithic Levant was composed of 48% Neolithic Anatolians, 52% Natufians, and then later on, much later, so if this is, we're talking about Neolithic Levant, let's talk about 8,000–9,000 years. The post-Neolithic Levant was also composed of 15.8% Neolithic Iranians. So basically, the Levant is made up of these 3 major components, okay? The Natufians, the Neolithic Anatolians, and the Neolithic Iranians. And the difference between this is the fact that what happened with Arabia, and I will show you in a minute, is, let's actually show you right now. What happened with Arabia is that this remigration into Arabia must have happened through 2 routes. The first route that came through Yemen, through the Gate of Tears, right here. And then this same route of migration is the one that went through Oman and all the way to Iran, okay, without any admixing with the Levantine population. And then the Natufians that actually populated the Levant, they split early on, and they moved into Saudi Arabia, and that's where the admixture happened. And that's why we see this difference between the Natufians that are the genetic of the Natufians that are in Arabia are very different from the Natufians that will be found in the Levant, just because there is Anatolian admixture and some steppe admixture into these Levantine. So basically, this is what we think happened, and this is what we think happened in modern populations that we know of are derived from these path migrations. As I said earlier, this is the first chapter of a long, process that we have not really touched upon the tip. What we are lacking, and it's something that is important, we are lacking ancient DNA from Arabia and the peninsula. And the problem, unless we have some DNA, ancient DNA from ancient Arabia, we may not be able to answer these questions the way we want. We think that modern Arabian populations may not have to do a lot with ancient populations, just because ancient populations may have been now underwater, have not been discovered yet, because of course the peninsula was a lot different back then than it is today. So I'm still hoping that we will unearth some DNA evidence, ancient DNA from the Arabian Peninsula, in order to really understand what's going on. So I kept my time. My beeper is beeping right now. My acknowledgement, and I thank you very much for paying attention.
Thank you very much, Pierre. Do you hear me?
I can hear you well. I can hear you well.
Thank you very much, Pierre. I would like to thank you for your contribution. Our second speaker is Professor David Comas. He's the head of the Human Genome Diversity Group in the University Pompeu Fabra in Barcelona. He is known for his research in genome diversity that modeled the present human genome variation. And his present topic is on the genetic insights into the population history of North African groups. So we finished, I think, with Pierre on the Arabian Peninsula on the east, and I think we're moving west. We're moving west now into North Africa. So, Professor David Comas, you're welcome to start your discussion with the group. Thank you.
So thank you very much, Professor Bayoumi, for the nice presentation. I'm going to share my screen with all of you. And I hope you can see now the screen. Okay. So let me check. Yeah. Can you see my screen? That. Yep. Okay.
Yes, we can see your screen.
Okay. Thank you very much for the. So thank you very much for the invitation. It's a pleasure and an honor to be here among you, and also it's a pleasure to share this session with Pierre Zaloua, who is a good collaborator and a great friend of mine. So what I would like to present during this talk is just an overview of the things that we know about North African groups, because North Africa, it has been usually neglected and avoided or forgotten in some of the genetic studies, and that's why we wanted to explore and to shed light into the population history of North African groups. So North Africa, despite being in the African continent, has had a very different history. And as I told you, most of the efforts done in Africa, they have been focused in sub-Saharan Africa, and North Africa has been usually neglected. It is known that the region of Africa has been populated since very ancient times, and in fact, the more ancient remains in Africa are found in North Africa, in this site in Morocco, dated around 300,000 years ago. But after that, after this first initial colonization, other cultures have inhabited the region. The Assyrian culture, we know that it's even older than previously thought, and other cultures like Ibero-Mauritian or Capsian cultures have been present in the region until the arriving of farming from the Near East with the Neolithic Revolution. And also posterior historical movements have had a lot of impact in the region. So one of these movements is the Arab expansion, and we know that nowadays the the speakers of the region, they are Afroasiatic speakers, and there are two main groups of languages, Arabic and Berber languages nowadays in North Africa. So our initial hypothesis is that North Africa was populated long time ago as a result of an old colonization with subsequent migrations. But there are still a lot of questions to be answered. So, for instance, is there a continuity since these first inhabitants of the regions with the present populations in North Africa? Or this ancient colonization was erased, wiped out by subsequent migrations in the region? Another question is if there is a genetic substrate autochthonous to North Africa, because sometimes North Africa has been presented like an admixture of components and external components from sub-Saharan Africa, the Middle East, of Europe. But what we wanted to address if there is a genetic substrate autochthonous to North Africa. We also wanted to address the role of the geographical barriers to this gene flow, the role of the Sahara Desert or the Mediterranean Sea, in order to try to address when this gene flow, this admixture took place. And the last question is to try to address if there are differences, genetic differences between Berber and Arab speakers in nowadays North Africa. So the first studies done in North Africa are based on classical markers, basically blood groups and serum proteins. And in this compilation that I show you in the screen, so these authors showed that the genetic diversity of these classical genetic markers had a clinal west to east differentiation. But most of the studies that we have done in North Africa are based in uniparental markers, the Y chromosome and the mitochondrial DNA. And as a summary, what we know about these markers is that there are in North Africa, there are lineages of sub-Saharan origin, there are lineages from the Near East and Europe. So these give us an idea of a mixture in the region, but also we can see some lineages that are autochthonous to North Africa, pointing to a genetic substrate that is original and autochthonous to this region. Nonetheless, as I told you before, so North Africa has been neglected and ignored in the genetic landscape. And here, for instance, in the Human Genome Diversity Project, in this analysis of more than 600,000 SNPs in these 1,000 individuals, only one population, the Mozabite, a Berber group from Algeria, was included in this panel. And as you can see in this tree, the basal groups are the sub-Saharan Africans, and they are distinguished from the rest of populations that are from out of Africa, with exception of the Mozabite, this North African population that lies in the base of this out of African group. But as I told you, at that time, there were very few data related to North African groups. And for this reason, so we wanted to fill this gap and provide data on North African groups. For this reason, so we analyzed some time ago several populations in North Africa, analyzing almost 1 million SNPs in several individuals, and also comparing these results with results from neighboring populations, and including in this first analysis one single population of Berber-speaking Tunisians. Okay, so what were the first results that we saw in the first analysis? Here in this multidimensional scaling plot that measures the genetic distances between individuals. Here each dot represents one individual, and different colors represent different populations. So what we can see is that sub-Saharan African populations are distinguished from Near Eastern and European populations at the opposite age. And in between, in these bluish colors, are North Africans, showing this heterogeneity and in the intermediate position between Sub-Saharan Africa and Middle East and Europe. And also, if we add a third component, what we see is a distinction of this Berber group from Tunisia compared to the rest of North African groups. Okay. So we wanted to address this differentiation. So what we did was a clustering analysis, this admixture analysis. And here I represent every vertical line represents one individual, and each color represents a putative ancestral component. So when we are analyzing two putative ancestral components, what we see is a differentiation of sub-Saharan individuals compared to the rest of individuals, although in North Africa, we can see this influence of these sub-Saharan individuals. When we are adding more components to the analysis, what we see is that North African populations are a complex, show a complex scenario with a lot of components and mixed here. Okay. As a summary, so what you can see here in North Africa is this light blue color that is found almost exclusively in North Africa, and this is an autochthonous component from North Africa. As a summary, here I can show you that there is this blue component, light blue component, that we hypothesize that is this autochthonous North African component, but also in North Africa, we can see an admixture of other components, such as a Middle Eastern component, this green component in a gradient form. It's also found in North Africa, but also a European component, this purple component, and also some components from Sub-Saharan Africa shown here in orange and yellowish colors. So as a summary, what we see in North Africa is an admixture of components, but also the presence of an autochthonous component. At that point, we wanted to address, so if we were able to distinguish this component, this autochthonous component in North Africa, and try to date when this component appear and split from the rest of components that we see in North Africa. In order to do that, and in a very rough way, so we use the FST, which is a parameter to calculate population differentiation. So between these components, and we were able to estimate the time of the split of this North African component from the rest of components, from the Middle Eastern and from the European component. And so we were able to date this autochthonous component more than 12,000 years ago, so previous to the Holocene, times. This was done taking into account the global ancestry of the individuals, but we can go a step further and instead focusing on the local ancestry of the individuals. Here, I present you the example of one individual, the chromosomes, all the chromosomes from one individual, and different colors represent different ancestries. So fragments of different ancestries within these individuals. Okay, this is very helpful if we try to estimate the admixture of these components in the population. So in this simple cartoon, let's imagine that we have two populations, the red and the blue population, and some red individuals go to the blue population. So at the beginning of the admixture, so what we will have is these chromosomes, red chromosomes into the population, but through recombination and through time, so we will see that these chromosomes are getting shorter and shorter. So if we are able to recognize these fragments, recognize the length of these fragments, so we can estimate the time of the admixture event. Okay, so we did that for the fragments of sub-Saharan ancestry, and we calculate the time of the admixture in North Africa, and we estimated that this admixture of sub-Saharan Africa into North Africa most of these events were very recent in historical times and maybe linked to the slave trade through the Sahara Desert. But at that point, so we didn't want to stay only with the gene flow to North Africa, but also try to address if we were able to detect gene flow from North Africa outside. And in this analysis, what we did was to compare fragments of North African ancestry in Europe. And what we saw was that in mostly all the North African influence in Europe was detected in Southern Europe, basically in the Italian peninsula and the Iberian Peninsula, and also in the Canary Islands, and that this gene flow was very recent. We were even able to date when this influence of North Africa into Europe took place. And when we focus on the Italian peninsula, taking a group of Tuscans as a reference, so we saw that the North African influence was a Mediterranean North African influence that took place in the area in Italy around the 5th century, so which is in agreement with the fall of the Roman Empire. So when we focus on the Iberian Peninsula and doing more or less the same analysis, we also saw that the North African influence was an influence of also the Mediterranean, and this was dated around 1000, the year 1000, which is also in agreement with the Islamic presence in the Iberian Peninsula at that time. And finally, we also estimate the presence and the mixture of North Africa into the Canary Islands. And what we saw here was a different pattern. So the influence was not an influence of Mediterranean North Africa, but the part of the Atlantic coast. And the date of the admixture was around the 16th century, also in agreement with the arrival of the Europeans into this area. But as I told you at the beginning, so in our initial analysis, we only included one sample, one Berber sample from Tunisia. And so we wanted to go beyond and to add more Berber samples in order to see if there was any structure, genetic structure in the region. And for this reason, so we add another Tunisian Berber sample, one Algerian Berber sample, and 2 Moroccan Berber samples in the analysis. And as a summary, so what we saw in this principal component analysis, where you can see the sub-Saharan individuals here, the Middle Eastern and European individuals here, and all these color-dotted symbols are the North African individuals. As you can see here, so there was not a clear cluster separating Berber speakers and Arab speakers. So what we see in general is a large degree of heterogeneity in the region, and there are no differences, no clusters between Berber and Arab groups. So we can see in the region this heterogeneous pattern, and when we are focusing on the mixture events, so we can see that around at that time, so we see that the Arabization had an impact in the region, but also other more recent events also took place in the region and modified all the genetic composition of the populations in North Africa. So at that point, and as Pierre previously suggested, so we also analyzed not only the diversity in single SNPs all over the genome, but we focus also in the analysis of complete genomes in North Africans. And also we compare these complete genomes of different regions in North Africa with already published ancient DNA from North Africa. In this case, these triangles represent the ancient DNA samples, and you can see that that we have data from around 15,000 years ago from this site of Taforal in Morocco, that is a late Paleolithic sample, but also we have some Neolithic remains, genomes in this area. And finally, we also compare this our modern data, present day data, with a sample of wenches in the Canary Islands. The first thing that we saw in a maximum likelihood tree is that all sub-Saharan genomes, all European and Middle Eastern genomes, and all East Asian genomes clustered together, and the North African genomes are spread across the tree, just again pointing to this heterogeneity and this admixture in these North African populations. And here in this principal component, again, the sub-Saharan Africans, the genomes of the sub-Saharan Africans are separated from the rest, but here, so there are all the European and Middle Eastern individuals, and in between these colored dots are the North Africans. I want to address your attention to these small triangles that are the triangles that I presented before, these ancient remains in North Africa. So as you can see, they are close to North Africa, but they are not on top of them. Okay? So there are some differences in these early sites in North Africa and the present day genomes in North Africa. With the genomes, so we were also able to estimate the effective population sizes of these populations, and here from the present to the past. And here what we can see, every line represents one genome and one population. And what we can see is that after the out of Africa, so we see that that sub-Saharan African populations, they had larger effective population sizes, and Europeans and North Africans, they have small, very little population sizes compared to this sub-Saharan Africa. We also calculated runs of homozygosity of these genomes and compared these sub-Saharan Africans, where they had smaller runs of homozygosity compared to Europeans and Middle Easterns here, and in between again, all North African genomes. And here, and this is the last result that I would like to show you, is the admixture analysis that we did with these ancient genomes, with modern genomes data. And I'm going to focus here in order to zoom in. And what we see when we compare these ancient samples to the current samples, to the current genomes, so what we see is that as shown before, we see a complex pattern of admixture in North Africa, but I want to drive your attention to these ancient samples in North Africa, these late Paleolithic and early Neolithic samples represented here in this orange color, that we see that this component is still present in the populations, in the present populations in North Africa. Which means that although different migrations, different gene flows have affected the region, have affected North Africa, they were not able to erase completely the substrate, the genetic substrate that it was present at least in late Paleolithic times. Okay, so we see still a continuity since late Paleolithic into the populations present nowadays in North Africa. And in all the simulations that we do in order to understand the genetic composition of North Africa, so on all of them, so we have to take into account some sub-Saharan influence, but also this influence from these ancient samples, which implies a continuity in the region. Okay, so as a summary, and in order to answer the questions that I first proposed, so we see, as I told you, a continuity in the region, a genetic continuity. We don't know if this continuity is from Archean times, but at least from the late Paleolithic, we see that the populations present nowadays in North Africa, they have had a continuation, a genetic continuation from this time. So we also detected a genetic substrate adaptogous to North Africa and differentiated from the rest of components of neighboring regions. So the role of the barriers, the Sahara Desert and the Mediterranean Sea, they have been permeable to the genetic flow between North Africa and neighboring populations. And most of these admixture processes took place in recent times, and we were not able to see large genetic differences between Berber and Arab speakers, and what we see is a large heterogeneity between groups. And to finish, I would like to thank all the people that have been collaborating in my group during all these years and participating in these works. Also, I want to thank the collaborators and the volunteers that participated in these works, and the multiple collaborators such as Pierre Zalua, Asmahan Bekada, Karima Fadlaoui, Zid, that they have been extremely cooperative, and it has been a pleasure to collaborate always with them. And also I would like to thank you for your attention. Thank you very
much. Thank you very much, David. I think it was an interesting talk about North Africa, combined with what Pierre has actually presented to us about the Levant and Arabia. We have now time for the questions. There are 6 questions on the panel, which you can view if you press on Q&A, and we can go through them one by one, because I think we have about 15 minutes to look at these questions. And I think whoever would like, maybe either you, David, or Pierre, you can come in at any time to answer the question. Now, the first question is, can you comment on the isolation of DNA? Sorry, I think I went into, let me just go into the first question. The first question is there any change in the size, perhaps also I add, on the sequence of the Y chromosome over the current chromosome sequence? I mean, this question is about any changes in the Y chromosome during evolution over the last 50,000 years.
Well, I don't think we've looked at this carefully, but it's a tough question to answer, actually. I don't expect to see much difference. 50,000 years is not a long time in terms of evolution. We see changes, perhaps, but to be able to notice size difference, I would doubt that. Dave?
Yes, yeah, so,
right. Okay.
The Y chromosome is very polymorphic in size within human population, so it's quite difficult to address this question. Yeah, but we have to take into account that the length of the Y chromosome is variable among humans. Yeah.
Thank you. The next question is, how did the heterogeneity within the Neolithic core zone affect the current population distribution in the Middle East and North Africa population? I think it's combined with another question, which says that what limited the spread of North African population groups to the rest of Africa? You mean going south rather than coming up from the south to the north, the direction of migration?
Yeah, so at least in North Africa, what we have seen is that the Sahara Desert, and Pierre mentioned that in his talk, the Sahara Desert has been greener and wetter in the past. Okay, so it has not been acted as a strong barrier to gene flow. And we see and we detect. I have been focused in North Africa, but if we focus in sub-Saharan Africa, we also detect gene flow from North Africa into sub-Saharan Africa. So which means that the gene flow was bidirectional, so north to south and south to north. Okay.
Right. The third question is, can you comment on the isolation of DNA from Egyptian mummies? If that has been done, and how useful is that in tracing the history of humans in North Africa?
Yeah, so this has been done. There are some data on Egyptian mummies, and I guess Pierre has more data than me on that. But I think it's very useful. I mean, ancient DNA samples now are key to understand the population history and evolution of our species, and these are very valuable tools in order to compare with modern DNA. Yeah.
Could I follow that with the question, next question, and maybe Pierre, you can answer this. Could Egypt be considered as a North African country, or is it an Asian country?
Yeah, well, Egypt is fascinating. It's absolutely fascinating. Egypt is a hugely diverse, population. I mean, it's a country that is of multiple countries. You have to remember that Egypt sits at the gateway between Asia and Africa, and then the if you want to talk about Northern Asia. So it's a fascinating country. And the problem with Egypt, I'll tell you, it has not been studied very extensively. So we lack a lot of data from Egypt. So whatever David and I have on Egypt still is extremely limited in a way that it doesn't tell us the full story about Egypt. I think Egypt holds a key to a lot of these questions that everyone is interested in answering. So yes, we see a lot of Asian DNA in Egypt. We see a lot of African DNA in Egypt, North African. So it's a total mixture, absolutely. And I've been focusing on comparing and contrasting Egypt and Yemen, because they both sit at the gateway from Africa, and very drastically different, extremely different, whether we talk Y chromosome, mitochondrial DNA, and even autosomal DNA. So massive project there.
Okay, can I also now give you the next question from Salim Bastaki, and he is asking whether the Berbers are descendants of the Vandals who moved from the east of the river Rhine to North Africa via Spain.
No, I think we can discard this hypothesis. I mean, the Berbers. Okay. I mean, they are completely genetically they fall into the North African context and landscape, and nothing to do with the Rhine Valley or the Vandals and things like that. I think we can rule out this hypothesis.
Yeah, I think I just like to go back to I think Pierre, is that Yemen is also very peculiar in the sense that Yemen is probably one of the refugia which during the glacial age, people still remained at the rim of the Arabian Peninsula and did not actually move out from Arabia. And that's why they're very distinct in many ways, in their Y chromosomes, in their mitochondrial chromosomes, and also in their lactase resistance genes. Okay, now the next question is, do you think that mitochondrial DNA have a role to play in population genetics?
Well, I mean, everything started by, the late Alan Wilson. That's how, this whole field exploded, is because of mitochondrial DNA. I don't think we can discard it yet. Absolutely not. I think there's a lot of information that mitochondrial DNA gets, especially ancient DNA. It's very difficult to extract DNA from ancient remains, and mitochondrial is the easiest. So absolutely.
I completely agree.
I think that paper came out in 1987, the Out of Africa hypothesis, and it is on mitochondrial DNA.
Yeah.
Mm-hmm. Right. The next question, I'm the question from someone who said, I am the principal clinical investigator of our microbiome signature. Can we combine it with our genetic signature?
Absolutely.
No.
Absolutely. We've been trying to do this now for some time. This is absolutely.
And it's amazing the interactions that we will discover between our microbiome and our genome, for sure.
Right. Nidal is asking, she said, according to my work on polycystic ovary syndrome on Sudanese families, we have SNPs that linked to Rhoda population. How can I follow them through African ancestors? I'm not sure about the Rhoda population.
Mm-hmm. And neither do I.
I think I may have heard of, maybe the spelling is wrong. You know,
Rwanda?
No,
I can't.
It's very easy. It's very easy. I mean, Africa has been extensively studied, and the 1,000 Genome Project has amazing data on, so she can really look that up easily.
All right. The next question is, which test was preferred more? Is it the mitochondrial DNA or is it the chromosomal DNA? Why? I mean, it probably, yeah.
That depends. That depends on the question you want to answer. I mean, if you want to explore your maternal lineages, so you have to go to the mitochondrial DNA. If you want to explore your paternal lineages, you have to go to the Y chromosome. And the information is I mean, it's you can add both informations to the genome. So I don't see that one or the other is better.
There's a question to you, David. I think it said, I would like to ask about the date of admixture you observed in North Africa. You dated that to the 13th century CE, connecting it to the Ghana Empire. So does this date mean that there were no past migrations from sub-Saharan Africa? Although we know that the Sahara was much greener than recent times, also populations from West Africa migrated north. Does this also confirm the distant clustering of the DNA samples from modern data on your PCA?
Yeah, for sure. I'm convinced that there were ancient migrations or ancient gene flow from sub-Saharan Africa into North Africa. But the methodology that we are using, and also the amount of the impact of the recent migrations, have been greater than past migrations. Yeah.
Next question: the population denoted as Yemeni, is it a collection of countrywide groups or is it a specific geographical site?
Well, Yemen is a complex country because it's tribal area, and there are they refer you have several. So the collection we have from Yemen is extensive. It's from various.
I think we're having sorry, I beg your pardon here. We're having sort of cacophony from somebody. I appreciate if people would mute their sort of mics for the time being until we finish the discussion of the questions. Now there is sorry, Pierre, can you continue, please?
So yes, the collection we have is actually from various regions of Yemen, trying to represent the various tribes, but you cannot catch it for sure. But we have a very wide and representative collection of Yemen.
Yeah, I think we also had to collect Yemeni samples from the capital in Sana'a, and there were different groups, people from different. There are 7 different sort of locations within Yemen, and you can't really tell who is who. There is a question from Afaf al-Sheikh. She's saying, are there any studies from Sudan?
They are, yes. Yeah, some studies. So not a lot of data, but there are some data. There is some data from Sudan.
We have a collaborator, actually, David and I.
Dr. Muntasir?
One of them is Dr. Muntasir, but there's a lady. If they're interested, if they can email me.
Yeah, I think I probably know them. The next question: is there any genetic knowledge about celiac disease in Saudi Arabia and Central Africa? Is there a connection between the two?
I don't have this data in my mind.
Okay.
I don't know.
All right. Next question. Does the high number of possible SNPs on the loci in the GWAS a factor that can increase the reliability of the results from GWAS compared to mitochondrial DNA or the Y chromosome data?
Absolutely.
Yes, for sure. For sure. I mean, the greater the markers that you are analyzing, more refined the studies you can perform, for sure. Yeah.
And the last question is, do you think consanguinity would impact population genetics and the waves of migration that happen?
Yeah.
Well, yeah, I mean, there's something we use. I mean, we correct for that. We do correct for that. Yes, they impact population genetics, but we correct for that. We have effective population sizes. You know, I mean, there's a lot of tricks that we do, and David is excellent on this as well.
Thank you very much.
Pierre.
Well, thank you very much, Pierre and David, for an excellent presentations and an excellent discussion that we had now. And I think I would refer back to Sami for some of the sort of housekeeping information that we need to feed back to our participants. First of all, we need to have your feedback. Please, for all those people who attended, and I just like to say that we had 478 people who attended this webinar, which is a very large number of people all over the world, which is a great feat for the type of webinars which CAGS is running. Now, we need your feedback. Please fill in the short survey that appears at the end of this session. The CME certificates will be downloaded as of tomorrow from the link. And I think, again, Stephanie has actually put that to all of you so that you can actually get your certificate. Our next webinar is on gene therapy, and it will take place on the 26th of May. And if you would like to register, follow the same steps that you have done for this webinar. And it is on the website of the CAGS, and you can see it also. I think it will go to all of you through an email address. Stephanie, do you have anything else to add?
No, that's it, Professor Riyad. Thank you very much.
Right. I just like to thank again David and Pierre for excellent presentations and excellent discussion that we had today. And I thank all our participants and the people who attended and their excellent type of questions and the active participation in a group like this. Thank you very much.